Hello, everyone, and welcome to today's broadcast, Quantifying microRNA and Protein Biomarkers Directly from Challenging Sample Types with FirePlex Technology. It is presented by Brendan Collins, Field Scientific Specialist for the Eastern U.S. for Multiplex Assays at Abcam. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Abcam. For more information about our sponsor, visit www.abcam.com. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Brendan. I will now turn the presentation over to him.
Thank you so much. Hello, everyone. My name is Brendan Collins. So I'm the Field Scientific Specialist for Abcam when it comes to our multiplex assays. Our multiplex assays are a brand name of FirePlex because due to the FirePlex particle technology that we utilize in these assays. And I'll take you through that in a few slides. So what I'm going to talk about today is quantifying microRNA and protein biomarkers directly from challenging sample types while using this FirePlex technology. Today, I'll take you through a brief overview of the FirePlex technology and the particles and how this works. Then I'll take you through our different assays that utilize these particles. I'll take you through our standard FirePlex microRNA and immunoassays. These allow you to multiplex up to 75 analytes in a single well. And then I'll talk to you about our brand new no-wash high-throughput immunoassay. This is a high-throughput. This is used for high-throughput screening and is automatable. So this is an introduction to the FirePlex platform. The FirePlex platform is a very new technology. It uses optical liquid stamping of hydrogels to create a large particle, which is a rod-shaped particle. These are then conjugated to either antibodies or probes to capture either proteins or microRNAs in your wells, your samples. So they're designed specifically for biomarker discovery and verification. With the microRNA assay, you can profile directly from biofluids. So this means no RNA purification. So you can directly profile microRNA present in serum, plasma, exosomes, urine, cell culture supernatant, cerebrospinal fluid. We even recently have launched a direct from tissue, formalin-fixed paraffin-embedded tissue, and cell suspensions. For the protein assay, the protein quantitation assay, the immunoassay, you can directly profile proteins in samples from cell culture supernatant, plasma, serum, urine, CSF, and bronchial alveolar lavage. This is, again, biofluids. Here's a picture of the particle. So the particle is a large, rod-shaped hydrogel particle made of PEG. So it's biologically inert. It allows for optical thermodynamics because it's sort of like a sponge. So it allows samples to move throughout the particle and get optimal binding throughout the particle as opposed to just on the surface of, say, a bead. It's inert, so you won't have other binding, non-specific binding to the particle from your sample. Each end is fluorescently labeled with a dye, and these are what create codes, if you will. So the codes are different levels of intensities of yellow and green, and that corresponds to, each code specifically corresponds to what is conjugated in the middle, which would be either a specific antibody to a specific protein or a probe specific to a microRNA sequence. And this is how you can get to 75 in a single well, because there are 75 different codes. It's then read out on a flow cytometer or a high content imager. The flow cytometer is what our standard assays use. to read out, and it runs through the cytometer single file, and then you end up getting 3 events. And then in this high content imager, it takes a picture of the well, and you'll end up seeing the different codes and the different intensities of yellow, green, and red. And I forgot to mention earlier, the middle region is read out as a red dye because we add a red reporter, which I'll take through in a few slides, the assay workflow, and that'll make more sense there. The very light gray regions are just blank. This allows spacing between the 3 fluorescent dyes, so your cytometer can distinguish between the 3 events. We also provide a free of charge software. And so what you end up doing is you take your FCS file from your flow cytometer or your TIFF file from your high content imager, import that into our software, and that will decode the particles corresponding to which specific analyte, and then it'll quantify these for you or normalize the data. This is a screenshot of our microRNA assay software. This is a heat map, so this shows differential expression of microRNA in different sample types, depending on which microRNA is in the panel. You can organize your plate layout here. You can create heat maps, bar plots, do all sorts of statistical analyses here. We actually can create publication quality figures with our software, and we have had dozens of publications with our assay. This is a screenshot of what the Immunoassay would look like. So these are standard curves, which you can designate on the plate, and you can quantify your proteins in your samples this way, much like an ELISA. We also offer a service lab in Boston. This is really useful for someone that may not have a cytometer or a high content imager. You can send us your samples. We'll do the services for you. We'll run the full assay. We typically turn projects around within 15 business days, maybe another week or 2 if it's a custom panel, which we do offer, and I will get to that later. But we not only just do the assay and send you the data, we send you a detailed report of what this data actually means, and we're happy to talk with you after the project is done to go through the data with you. So now I'll take you through our FirePlex microRNA assay. This is the assay overview. So it takes about a day to run. So you start with your tissue or your biofluid. With biofluid, you can work from 20 microliters of sample input. This is really useful if you're working with mouse or rat, and you may not have a lot of sample to choose from. So you add our digest buffer to the sample, and that will release the microRNAs in your sample. And then you add panel, which contains the particles with the specific microRNAs that you're looking for in your sample. After incubation of the panel, you hybridize, and you're actually going to elute the bound microRNAs off of the particle, of course, after you wash. After you wash, you elute it off, and then you're going to run PCR. And I will take you through the molecular workflow in the next slide. So with PCR, you're amplifying, and then at the end of that, you actually have a biotinylated complex, and you're going to end up transferring it back to the plate, recapturing onto the same particle mix you had before, and then add a reporter, which is the streptavidin PE-Cy5 to give you the red dye, the red readout, and then you can scan it in your flow cytometer and analyze in our software. This is the molecular workflow, so it's really helpful to see how this actually works from this level. So these are different particles, as you can see. The blue region is where the probe is conjugated to. There are millions of binding sites here. The light gray regions on each end of the probe are what we call the universal label region. These are the same sequences throughout the particles, no matter which microRNA they're trying to capture. Whereas the blue region in the middle, this is the target-specific region. So this is a sequence specific to the microRNA you're looking for. So you add your sample, and this probe will capture microRNA 16, another one will capture microRNA 24, et cetera, and up to 70 different microRNAs in the sample. And then after you have the microRNA bound, you're going to wash, and you're going to add a universal label mix. These will bind to each end of the universal label regions. One of these labeling mixes has, oh, sorry, I got a little bit ahead of myself. Once you have the complex, which is the universal label, the captured microRNA specific to what you're looking for, and then the universal label on the other end, it creates the complex, and then you elute it off and you run PCR. And you're going to run PCR with universal primers against these universal regions, not the target specific region. So because these are the same universal regions, regardless of which probe they are, it'll amplify equally across all of the microRNAs. And on one of these primers, there's a biotin. So that allows you to add the streptavidin P5 dye to get the fluorescent readout. So once you've amplified, you have your biotinylated complex of microRNA universal label. You capture it back on the same particle mix you captured earlier in your samples, and then you add a reporter to get the fluorescent readout, and then you run it through the flow cytometer and import it to our software. So this is an example of a heat map of a panel that we used on different sample types. So this is a 65-plex panel capturing different microRNAs and different sample types. You know, these are highly expressed, these ones are lower expressed. But as you can see, with plasma EDTA, plasma heparin, serum, these all worked really well and very consistently. We've also worked with amniotic fluid, exosome preps, saliva, formalin-fixed paraffin-bedded tissue, and even purified RNA if that's what you are working with. So for sample input, for biofluids, we only require 20 microliters. For tissue sections, we only require 5-micron curls. Again, none of this requires RNA isolation ahead of time, or cDNA preparation, or pre-amplification. You can work directly with your samples, which makes it very easy to work with. And if you have purified RNA already, that's totally fine. You would just skip the digest step and work right with the panel, capturing the microRNAs of interest. So I'll take you through some data of how we validated this assay. So this is how we've determined the 20 microliter of sample input based on the sensitivity of the assay. So we started with the custom 48-plex panel. And when we added pooled human serum, we added 40 microliters and 20 microliters, 10, 5, and 0 microliters. We saw no difference between 40 microliters and 20 microliters of sample input. These captured on average 35 microRNAs of the 48. And the other 13 just mean that they weren't present in the sample. And then when we added 10 microliters of sample, it only reduced it down to an average of 29 or 30. So we lost a few microRNAs that were lower expressed, but you can still go down to 5 and still capture 15 to 20 microRNA. So this is where we determined that 20 microliters is a really good input amount. It really makes no difference to 40. And then it saves you sample. It allows you to run a duplicate if you only have 50 microliters. And over here, this is brain, lung, and liver reference RNA, and this just shows that we are able to capture microRNA and lower inputs of picograms of RNA. This slide shows the specificity of the assay. So we are able to distinguish single nucleotide discrimination within families and closely related microRNAs. Here we had two panels. One was a LET7 family and one was a 302 family of microRNAs with very small differences of sequences. We were still able to distinguish relatively. We were still able to specifically distinguish these microRNA from the other. And of course, some are higher because it really depends on where those changes fall on the molecular level. Like if they're on the end of the sequence, they're still going to get binding and you might have a floating tail there. But overall, it's very specific. This slide shows the reproducibility. You know, we've done the assay at different days, and we were able to show that they still strongly correlate depending on what day you run it. The panel on the same samples. And this slide shows that we are able to profile in samples containing inhibitors without any issue. So a lot of times you might have samples, plasma samples with heparin or sodium citrate, where that might not work because of the inhibitors in those samples. But due to all the wash steps that we have, before you even before you even run PCR, you're going to wash the samples to wash away any of the inhibitors in those samples. So by the time you're actually running PCR, those are all gone. And this slide is a platform comparison of the FirePlex assay versus tilde cards in Illumina TruSeq. We have strong correlation between the three assays, and in fact, we have smaller sample input, and it just as strongly correlates. So now I'll take you through what we have to offer for the miRNA assay. So we essentially have three different types of panels that we offer. The first one I'll talk about is our discovery panels. So these identify miRNA signature in biofluid samples like plasma, serum, and cell culture supernatant. These are pre-designed panels of 400 circulating microRNAs. So these are 400 microRNA selected from peer-reviewed studies. We have a plasma serum focus panel, an immunology focus panel, and a rodent-specific panel for if you're working with mouse or rat. Soon, we're going to be releasing a C. elegans panel and a viral microRNA panel. So this is really useful if you aren't really sure which microRNA might be in your sample. You might have an idea of some, but there might be others you might not have considered. This actually requires a little bit more sample input because it's going to be split across wells. You can do about 12 samples per plate, but essentially you're going to get 400 microRNAs out of your sample. What a lot of our customers do is they'll do a small study with the discovery panel, and then they're able to see, these 35, these 40 microRNAs actually show strong differential expression. Now we want to study these further, and then we can design a custom panel for you, which I'll talk about. We also have focus panels. So these are smaller focus panels that are specific to different research areas like oncology, immunology, neurology, and cardiology. We also have different tissue toxicity panels for kidney, liver, pancreas, and cardiac toxicity. These are the 65 microRNAs in a single well. This would be with your 20 microliter of sample input. So these are more focused if you're specifically looking at this area of research. And finally, the best offering I think that we have is our custom panels. We can build a custom panel to any microRNA sequence that you're looking for. So we've had some customers do very small panels, screening across a large number of samples. We can go from 5 microRNAs up to 200. So if you're working with, a different species that might not have homology with mouse, rat, and human, we can build that as long as you have a sequence like canine, monkey, etc. If there's some novel microRNAs that might not be in any of our focus panels, we can build those too. As long as we have a sequence, or as long as if it's in miRBase version 21, we can design that for you. So here's an example of some custom microRNA panels that customers might use. They might want to design a panel for macrophages or dendritic cells or T cells. We have a really useful FirePlex discovery engine tool that I'll take you through in the next slide. Essentially what it does is it searches PubMed, and it searches all the abstract words and makes a word cloud of the most popular, most cited words in those abstracts, depending on which area of research you're looking for. So this is a word cloud where someone searched for macrophages, and probably human, and these are the most commonly cited microRNAs. And what's really useful is the discovery engine actually links you to those PubMed articles that you can actually click on to go read. Here's another example of custom microRNA panel that someone might design for organ injury. The link is down here of our FirePlex discovery engine. This is what it looks like. Essentially, it's just a little search bar like a Google. You type in oncology, you select the species of interest, you hit search, and then you get the word cloud. You'll also get oh, this is a screenshot of what it looks like. So this will be the word cloud here. These are the most commonly used cited words. These are the most commonly cited genes, and these are the most commonly cited authors. And these actually all are hyperlinks, so you can click on them directly. Now I'll take you through our multiplex immunoassays. The immunoassays are essentially ELISAs run instead of the capture antibody being on the bottom of your ELISA of your 96-well plate, the capture antibody is on the particle itself with the specific code corresponding to that antibody. In this assay, it only requires 12.5 microliters of biofluid input. This can multiplex up to 75 analytes in a single well. So you add your capture antibody. That capture antibody detects the protein of interest. So if this is TNF-alpha, it'll capture TNF-alpha in your sample. We provide the detector antibody, which is biotinylated, and then you add a streptavidin PE-Cy5 reporter dye, and you run it through the flow cytometer just like you did with the microRNA assay. And then you import that into our software for analysis. Here's the workflow details slide. So again, capture, wash, detector antibody, wash. You only need to incubate for an hour at room temp for each of these. You could also do an overnight incubation at 4 degrees if you want. We're pretty flexible that way. Then after the last wash, you add your PE-Cy5 and you run through the flow cytometer and you import to our software. And this will plot your standard curve. We provide the standard curve mixes. So if it's one of our 17-plex key cytokine panels, we would provide you with mix A, which has all 17 of those in a single mix. Here's a couple of our pre-designed human panels. We have an inflammation panel, key cytokine 17-plex panel, key cytokine 4-plex, and a Th1, Th2 response panel. We also have the same offering for mouse, rat, and non-human primate analytes. This is a screenshot of the dynamic range and the sensitivity of our top 17 key cytokine panel. So these assays all use our rabbit monoclonal antibody pairs. All of these pairs are produced in-house at Abcam. They're recombinant monoclonal antibodies. So because of that, they're highly sensitive. They have very strong lot-to-lot correlation because they're recombinant antibodies. They also have a really broad dynamic range, which is really useful. If you're trying to measure healthy versus sick patients, you could see strong inflammation spikes without it going over the dynamic range. Depending on the analyte of interest, about half or most of our analytes have higher sensitivity than our competitors. These would be the ones in purple highlighted here. This is based off of what our competitors have put online of their cytokine panels. And as you can see, we have 4 to 5 log dynamic range here for all of them in the same well, which is really useful. So instead of having to run 17 different ELISAs, you can just do one assay here, and it saves you a ton of time and a ton of money. We also have a 70-plex pre-designed panel. This was launched 72-plex, sorry. This was launched a few weeks ago. We've had a couple customers run it very successfully in our service lab so far. This is a list of the 2 36-plex panels. We have panel A and panel B, which you can get as well. But the 72-plex is just a combination of panel A and panel B. And these are a list of all the analytes you can profile in your sample, only 12.5 microliters. You can capture all of these proteins in a single well. And as far as we're aware, this is the largest multiplex immunoassay panel currently on the market. This is a screenshot of the dynamic range of the 70-plex panel. So despite them all being in one well, they still have really strong dynamic range here. And some of these have really low sensitivities, sub-picogram. We screen these in all sorts of samples. So we've screened the 70-plex in in plasma EDTA, heparin, citrate plasma, in cell culture supernatant, CSF, and urine, and serum. So we're releasing a 70-plex mouse cytokine panel as well. This should be, the ETA is October 2018, so coming up really soon. These should have very similar markers as the ones here in the 70-plex panel. We also offer custom immunoassay panels for, this is another example, where you could design your own panel for oncology research, say for cytokine release syndrome or immuno-oncology markers. If it's a subset of our 70-plex panel, since we've screened these pairs together and then we've validated these all together, we can just design you a custom panel of that. We're also releasing some organ injury and toxicity panels very soon as well. So kidney tox, cardiac tox, and a liver tox panel. Our simple step ELISA kits and our FirePlex assays have very strong correlation. In fact, most of the pairs actually are the same in the simple step ELISA kits as they are in the FirePlex assay. So if you were to screen, say, our 17-plex key cytokine, and then you're looking at you want to just look at TNF-alpha across more samples, you can get a simple step ELISA kit. It's the exact same pair that was used in our FirePlex assay. So the assay itself takes about 90 minutes to 2 hours to run, and then scanning on the cytometer. You can analyze up to 75 analytes per well from only 12.5 microliters of sample input. It's read out on a 96-well plate and then on a flow cytometer. The sensitivity, depending on the analyte, is for the most part sub-picogram per mil, and again has 4 to 5 log dynamic range and less than 50% interval CV, and is compatible on lots of biofluids like plasma, serum, cell culture supernatant, CSF, urine, and bowel. Again, this is the reason why this is so able to get 70 samples, 70 analytes in a single well and have such high sensitivity is because of the recombinant monoclonal antibody. Again, because they're monoclonal, there's low cross-reactivity across samples. It's recombinant, so you get low lot-to-lot variability, and you get high sensitivity as well. These antibody pairs are in our multiplex immunoassays, the FirePlex particles. The simple step ELISA kits. We also offer matched antibody pair kits. So if you want to scale up ELISAs, you can get matched antibody pairs. Now I'll take you through our FirePlex high throughput immunoassay. This is a brand new product launched in July. So this is for high throughput screening, and it's very similar to the immunoassay just discussed. In fact, it's a simpler workflow, which is key for high throughput. So you add a FirePlex reagent mix to your samples, so one of our panels to your biological samples. It only requires 6.25 microliters of sample input. So you're going to add 6.25 microliters of your sample to 6.25 microliters of the antibody mix, the panel mix. You're going to incubate overnight at room temperature, and then you're going to add a imaging dye. The imaging dye will quench any excess autofluorescence within the sample. So this is key for a no-wash high-throughput assay. The particles all then settle to the bottom of the well. The imaging dye quenches everything in your sample, everything not bound in your sample, any autofluorescence that might occur. And then you put it through a high-content imager, and it'll take a picture of the well. These are read out on 384-well plates, I should mention. So this is really useful if you're screening hundreds to thousands of samples. The reason why the high-content imager is really useful is because it only takes 10 or 20 minutes to scan the whole plate and take a picture, as opposed to a flow cytometer, which might take an hour and a half to 2.5 hours, depending on the cytometer, for a 96-well plate. So if you think 384 wells in 20 minutes or 96 wells in an hour and a half to 2 hours, this is way better for high-throughput screening. You take the image file and directly import it into our software, which is the same one you download for the microRNA and the standard immunoassay, and you do the same data analysis as you would before. So we validated a couple imagers so far. The Image Express for molecular devices, TTP LabTeX Mirrorball, PerkinElmer Opera Phoenix, and a few others. This is a really useful picture because I think it's really nice because you can actually see the particles, you can actually see the codes and the analytes captured with the red dye. So if you look closely, you can see different levels of green and yellow combinations with a red in the middle. And those are that's the particle ends, and those are the codes that correspond to the specific protein that's captured. The red is, again, the antibody that's captured the protein, and the different levels of intensity of those just implies the different quantities in that sample, of that particle in that sample. These are some of the analytes we've validated so far. So we've done about 30. So they still have pretty high sensitivity for a no-wash assay. Again, you can't compare to the standard immunoassay because of how many wash steps there are. You're going to get better sensitivity. But even with no wash, we're still able to get fairly low sensitivity while maintaining 2 to 3 log dynamic range. Here are some of the panels available. We have a human 10-plex, human 6-plex panel. These are key cytokines. We're also developing a interferon 6-plex panel that should be coming very soon. This is some of the panel validation. So this is the analyte performance. You can see that it still has, it still has 3 to 4 fold dynamic range. It has very low cross-reactivity. These are all, this is 15 to 20 different analytes. And they have very low cross-reactivity. And we did a spike recovery, and all of these show, for the most part, within 30% spike recovery. The high throughput assay is also designed to be automatable for those that want to automate and not, since it's, if you're going to be doing many plates in a day. So we are able to do this, and we're happy to discuss this with you. So for the high throughput immunoassay, again, so it's up to 10 analytes per well as opposed to 75 in the standard immunoassay. The reason for this is because the 384 wells are very small. You're adding much less sample, and the particles themselves are actually fairly large. And because you're taking a picture, you don't want them to be stacking on top of one another. Otherwise, you won't be able to the software won't be able to distinguish the codes. So we found that with the amount of analytes, with the amount of particles we put in per analyte, 10 is the optimal amount. But it's read out on a 384 well plate, so you can get much more samples out of this. It's read out on a high content imager, so it's much faster because it only takes 10 to 20 minutes to scan. Sensitivity is still 1 to 100 picograms per mil for a no-wash assay, which is very good. And again, 6.25 microliters of sample input. This is validated so far in serum plasma and cell culture supernate. We're working on other sample types too. Finally, I wanted to mention that Abcam also has custom antibody development. This is a flexible, full cycle support of custom antibody development. So you can consult with us. We can produce RAB-MABs for you. We can do NGS for you, phage display, etc. We have experienced specialists in antibody design that can support you every step of the way. You can even think about a custom antibody design for use in FirePlex as well. Thank you so much. I'm happy to answer any questions.
Thank you, Brendan, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Let's get started. Our first question is, does this work on tissue or cells?
Thank you. Yes, it does work on tissue and cells. It's actually recently been a validated sample type. So no need to do RNA extraction from tissue or cells. Our digest buffer will just digest the tissue directly and release the microRNAs, and then our panel can capture those.
Okay, thank you. Which flow cytometers and imagers are validated?
So we validated many flow cytometers. So we validated Guava and Tune, Accuries, LSR2, Cortesa, a lot of the BD machines. If you can reach out to us and ask if any of the cytometers that you have are validated. And we have a field application scientist that can work with you directly to help set up the cytometers or the imagers, and make sure that those will be equipped for you before you start the assay. And what we do is we have a particle, what we call a cytometer setup kit or a imager setup kit. These are particles without probe or antibody in them, but just the dyes. So you can run them through the cytometers or take images of those ahead of time before you use any samples or any of our panels to make sure that those are calibrated ahead of time.
Okay, thank you for that. Next, how much sample input does this require?
So for sample input for the biofluid assay for microRNAs, so for the microRNA assay, in biofluid sample input, it's 20 microliters. We can go down to lower amounts if needed, if you have less, but 20 microliters is optimal. For the immunoassay, the standard immunoassay, is 12.5 microliters. For the high throughput assay is 6.25 microliters.
Thank you. We have time for one more question. Do you have to extract RNA?
No, you don't have to. So again, you can just profile directly from the biofluid or directly from the tissue. But if you happen to have already extracted RNA, that's totally fine. It'll still work with our assay. But just if you have your samples ahead of time, there's no need to do that.
Great, thank you. I would like to once again thank Brendan Collins for his presentation. I'd also like to thank Abcam for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 27th, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.